Hey everybody, welcome to our Selective and Differential Media Lab. We're not going to talk about biochemical testing until next week, okay? In this lab, we're going to be learning about phenylethyl alcohol agar, mannitol salt agar, Eosin methylene blue agar, and citrate agar. So the first agar is phenylethyl alcohol agar. This is also known as your P plate. It is a selective media. When you say the term selective, selective is a media that inhibits the growth of certain organism and selects for the growth of other organisms. P plates select for the growth of gram-positives and inhibit the growth of gram-negative bacteria. We use this media to inhibit the growth of our gram-negative organisms when they are in a mixed culture. So say you are working with your unknown test tubes and you have ran a test, a gram stain, and you notice that you have gram-positive bacteria, but there's also some gram-negatives. To inhibit the growth of those gram-negatives and to isolate just the gram-positives, you can place the bacteria on the PEA plate. And so in our lab, each of you is going to receive 2 PEA plates. So each human will get 2 PEA plates. You're going to inoculate one of the PEA plates with a gram-positive, and you're going to inoculate the other with a gram-negative. And then we'll incubate them until Thursday. The next media is a selective and differential media, and that's known as your MSA plate or mannitol salt agar. So MSA is selective to organisms that can grow in salt conditions or organisms that are halotolerant. It inhibits organisms that can't grow in a salty environment. So it is differential in that those organisms that do grow will be separated based on if they change the media color or not. So if you look over here, this is a mannitol salt agar plate, and it is normally a red color, but the bacteria over here are utilizing mannitol. They're fermenting the mannitol, and that causes the media to change color. So we're not looking at the bacteria color, we're looking at the media color. This is what the media looks like when it's not being fermented. This is what it looks like when it is. So it's selective for salt tolerance. Selection is always first. And then it differentiates those that do grow based on if they can ferment the mannitol or not. So each individual will get 3 mannitol salt agar plates. And you're going to inoculate Staphylococcus aureus on one of them. You will inoculate on a second plate either Staphylococcus epidermidis or Micrococcus luteus, depending on what you have at your table. And then on the third plate, you will inoculate either Lactococcus lactis or Enterococcus facialis, again, depending on what you have at your table. And you'll incubate those inverted until Thursday. Eosin methylene blue is a selective media. I shouldn't say that. Eosin methylene blue, or EMB, is selective and differential. It is selective for growth of gram-negative bacteria. So remember, our P plates are selective for gram-positives. This is your gram-negative selector. It inhibits the growth of gram-positive bacteria. EMB is also a differential media. So it differentiates bacteria based on their ability to ferment lactose. So bacteria that don't ferment lactose will be clear. So they'll grow and they won't have any color. You'll see the growth though. Bacteria that do ferment lactose will change color. So the bacteria themselves change color. See how the bacteria is like a purple, but on top there's this darker purple? That's a lactose fermenter. And bacteria that are extreme fermenters will produce a green sheen. So we have a non-fermenter, a fermenter, and an extreme fermenter. So first, this media selects for growth of gram negatives. So all of these are gram negatives. Second, those that are growing are going to be differentiated based on their ability to ferment lactose. So each group, or no, I'm sorry, each, that says group, but I mean individual. So hold on, I'll change it. This looks better. Each individual will take 3 EMB plates. And on one of those EMB plates, you're going to aseptically transfer E. coli. On the second, you're going to aseptically transfer Shigella. And on the third, you're going to aseptically transfer either Staphylococcus aureus, Staphylococcus epidermidis, or Lactococcus lactis. So one of the 3, depending on what you have at your table. You'll incubate until Thursday, inverted. And the last test you're going to do then is you're going to run a citrate test. Citrate is a differential media. It differentiates based on the ability of bacteria to utilize citrate or not. So organisms that, so to utilize citrate in an anaerobic process or not, so in a fermentative process. So if citrate is able to be broken down via fermentation process, then the pH indicator, which is bromothymol blue, will respond, changing the pH to a more alkaline color, and it'll move from a green to a blue color. So this side shows citrate negative, and this side shows citrate positive bacteria. And so each group is going to, or each individual, again, man, I keep messing this up. Each individual will receive 2 citrate plates, will take 2 citrate plates, and then you will aseptically transfer E. coli on one of them and Salmonella typhimurium on the other. Incubate inverted until Thursday. And that's it for this lab activity. The other lab activity I'll talk about in a second video, which is going to be your colony morphology. Bye.